All right, so chapter 16 is titled The Molecular Basis of Inheritance. Pretty much, we're going to be talking about DNA. We're going to review the structure of DNA, talk a little bit about the history behind the discovery of the structure of the DNA, and then primarily the rest of the chapter will be looking at how DNA is replicated or copied during S phase of the cell cycle, right before a cell goes through cellular division, either mitosis or meiosis. So a little bit of background. Believe it or not, we actually didn't know what the genetic material in a cell actually was. There are a lot of different macromolecules in cells, and there were two main constituents that most scientists believed one or the other was going to be the molecular basis of how genetic information is passed on from one generation to the next. And the two main constituents were either proteins or nucleic acids. And the reason why scientists pick these two is because if you think about it, they're both built as polymers, and they have these subunits, these different building blocks that you can use to build really complex and very varied polymers out of those few subunits. Now, many scientists thought, well, you know what? It's got to be the proteins that store the genetic information, because if you think about it, there are 20 amino acids, 20 building blocks to use. That's going to give you the flexibility to create really varied molecules. Where others were like, yeah, I kind of get that point, but I really think that it's going to be the DNA. Yes, there are fewer building blocks. We've only got 4 different kinds of bases that are used in DNA, but still, with just those 4 and an endless number of these nucleotides that you could put together, you could also create an infinite number of these genetic information storing molecules. And so scientists had this question that they need to answer now. Is it the protein or is it the DNA in a cell that stores the genetic information? And so your book goes through some of the experiments that were done to determine that it's actually the DNA, not the protein, that stores the genetic information. So if you want more info, go read the beginning of that chapter. I'll go through the experiments just very superficially. So really, this was early in the 20th century where we identified that DNA were these molecules that house the genetic information. And the way the experiments were done was using bacterial cells and bacteriophages. Remember that bacteriophages are those viruses that just infect bacterial cells. And the nice thing about these bacteriophages is that they are just created out of 2 macromolecules. Their genome is made out of DNA, and then you've got the capsid that's made out of protein, right? These 2 molecules that are, going head to head on which one is going to be housing the genetic material. So pretty much what scientists did is that they labeled either the DNA or the proteins one at a time with radioactivity, and then they allowed these radioactively labeled bacteriophages to infect bacterial cells, and they followed to see what part of that virus gets into the cell and potentially takes the cell over as a hostage to make more bacteriophage particles. And so what the scientists determined is that when you labeled the DNA with radioactivity, the radioactivity made its way into the bacterial cell, and that kind of gave us this idea that the DNA is going to be that holder of genetic information that then will take the bacterial cell hostage, or that passes on genetic information to the next generation during cell division. All right, now once we determine that it's the DNA that holds this information, then we had to answer a lot of questions about the structure of DNA. And there were 4 scientists that were critical, amongst others though, but 4 that were the main players in terms of determining this double helical structure that we now widely accept as the structure of DNA. And these 4 scientists were Maurice Wilkins, Rosalind Franklin, James Watson, and Francis Crick. Now, the first 2 I mentioned, Maurice Wilkins and Rosalind Franklin, they were both X-ray crystallographers. This means that their science, the science that they did, their research, primarily used this technique of X-ray crystallography. Now, we haven't had a chance to talk about this in great detail throughout the course of the lecture class, but just to give you a little bit of background information, X-ray crystallography is a technique that scientists can use to determine or to get ideas of the structure of a molecule. What shape does it have? And the way this technique is done is you take your molecule of interest, maybe it's a protein, in this case it was DNA, and you purify it. So you have a very, very pure solution of this molecule, and then you force it into a crystal. Once you have a crystal of your protein or a crystal of the DNA, then you take X-rays and you shoot the X-rays through the crystal. Now, as the X-rays move through the crystal, depending on the shape and structure of the crystal itself, those X-rays will be diffracted. They're going to be bent, right? And they're going to go in different directions. And so then if you take a piece of film on the other side of the crystal, you can collect that information in which direction all the X-rays were diffracted. And you get a diffraction pattern. And one example is shown here in the lower right-hand corner of your slide. This is the diffraction pattern that Rosalind Franklin, pictured here to the left, developed for the DNA when she was performing this X-ray crystallography on her DNA crystals. And so she made this image and she had it lying around on the lab bench, and it just so happened that James Watson and Francis Crick happened to be visiting her lab. And they were walking by and they looked down, saw this image on the lab bench and said, huh, that's really curious. This molecule that you took this X-ray crystallography image of, it looks like it would be a double helix. What is this molecule? And Rosalind Franklin told them that this is DNA. And so Francis Crick, James Watson took this information back to their lab and they started thinking about it, right? So given the information here from the image that DNA is a double helix, how could that potentially fit together? How could we create this model? And so they worked on it for a while. Interesting thing, right? When I look at this X-ray crystallography image, I'm not an X-ray crystallographer. So to me, it looks nothing more like a Rorschach image and I can see whatever I can out of it. But to an actual X-ray crystallographer, they would take measurements based on the diffraction pattern they see. And they have the ability then to say that, yes, I think this molecule is double helical. It's pretty crazy. So anyway, back to James Watson and Francis Crick. They took this information back to their lab and eventually they pieced together, using Maurice Wilkins, Rosalind Franklin's data, other scientists like Chargaff, they used their information and they came up with this double helical model. And so here you see a picture. This guy right here is Francis Crick. This guy down here is James Watson, and they're standing next to their ball and stick model of the DNA double helix. And so once they published this in 1953, scientists widely accepted it, and the next step was really then to figure out, okay, we know the structure of this molecule, we need to understand a little bit more about how it's replicated and how it's actually used to store genetic information. And we do now know that our hereditary information, or the hereditary information all cells, is encoded in our DNA, right? It's found in pretty much every single cell of the human body, with a few exceptions, like red blood cells. They don't have a nucleus to begin with. So no DNA there. They don't last very long. This is why you're constantly replenishing your red blood cells. But for the most part, all other cells in your body are going to have DNA as their genetic information. And what we've learned is that DNA really directs the program of development. It controls biochemical processes, it controls your anatomy, physiological, and even to some extent your behavioral traits as well. And so the rest of the chapter, now we're going to review in a little more detail the structure of DNA, and then we're going to talk about DNA replication, right? This process of how do you copy the genetic information so that you can pass it on to the next cell, so that each daughter cell has the necessary information it needs to be able to function. All right, so a little bit of a review here on the structure of DNA. So remember, it's double helical, right? And this means that there are actually two DNA polynucleotide strands that are running parallel to each other, and then you twist the whole structure to make that double helix. We have covered earlier that the two strands of the DNA, or the two polynucleotide strands, they have this sugar phosphate backbone where you have the nucleotides attached to each other using covalent bonds between the phosphate of one nucleotide and the sugar of another. And then the nitrogenous bases, the A's, T's, C's, and G's kind of hang off to one side. And then you'll take one of these strands and you'll match it up in parallel to another strand, polynucleotide strand, except that remember, while they do run parallel, they don't run in the same direction, right? They're going to run anti-parallel. So the head of one is opposed to the tail of the other one, or what we call the 5' end and a 3' end. Just a little bit of review. Remember that the 5' end is called the 5' end because the sugar that's closest to that end of the polynucleotide, the carbon on the sugar that's closest to that end of the polynucleotide strand on the 5' end, this is the 5th carbon. So if we were to count the carbons here in this deoxyribose monosaccharide, this is carbon number 1, 2, 3, 4, and then this one's 5. So that's the 5' end. The opposite end, also counting the carbons in the sugar, this is 1, this is 2, this is 3, right? So this 3rd carbon would be added to the next nucleotide down here. So this is called the 3' end. Now, Watson and Crick also took other scientists' information indicated that there was a very specific base pairing pattern in DNA. So adenines always would pair with the thymines, and the guanines always pair with the cytosines. It doesn't happen any other way. Otherwise, the structure of the molecule wouldn't work. And so they said, well, that's really interesting. If you only have one strand of this double helical DNA, you automatically know what the other one is going to look like. Maybe this is what cells do when they replicate their DNA. Maybe they pull apart the two strands and use both of them as a template to make a matching polynucleotide strand. And so they suggested this as a possible copying mechanism for the genetic material. Watson and Crick, they themselves didn't get a chance to do these experiments, but the complicated experiments were performed by other scientists, and their hypothesis was actually indeed supported.